Awesome. Thanks everybody for coming out today. I'm uh, excited to introduce Sergey Ovchinnikov. It's a bit of a quick change in scheduling. Joe wasn't able to make it today, internet issues, so he'll come and present next month. But Sergey is going to tell us an exciting story about his latest ML work, which is posted in bioRxiv. And Sergey, yeah, take it away.
Awesome, thank you. Actually, let me, I'm just gonna add the link again, but I just included a tiny URL link, and what that will send you to is a Google Colab page where you guys could technically just run the code and start doing your own design directly in your web browser. But it also includes the link inside there to the slides. So if anybody wants to follow along with their own set of slides and keep notes, whatnot, feel free to make a copy of those. Also during presentation, feel free to interrupt me with any questions. Not a problem. Either just turn on your mic and just start yelling, or you could also put in the chat box. Either way is fine. Actually, maybe turning on the microphone might be better, because I think when I make it full screen, it doesn't always alert me that somebody sent a message.
So again, I'm not sure there's any permissions issue on that file. I can't grab it. I don't know if anybody else is having.
Oh, really? Let me make sure. It says anywhere on the internet should be able to view it. Actually, no, let me send also the, let me send the non-tiny URL. Maybe there's an issue with the shortcut that I made. Try that instead. Actually, let me also send the slides just in case.
I'm still not an error, but I don't know if other people are.
It seems like there's other people online, so I think it's probably well, I mean, I can also send you an email later.
Yeah, no worries, no worries. I just
okay. All right, so I'll go ahead and start sharing my screen. Yeah, so thank you very much for coming here last minute. I apologize if some of these slides may look a little redundant. I've presented some of this stuff in the past, including the TR Rosetta work, but since there's quite a few new people, I thought it might be good to reintroduce some of those concepts. I've been recently involved in a few side projects with David Baker's group, where we try to take some of the work we did with structural prediction and flip it around to do protein design. And today I'll be mostly focusing on this work here, where we say design a new sequence for a given backbone. But if we have time, I will also touch upon some of this other work that was mostly led by Ivan Nishenka on seeing, can we actually not only just design a new sequence, but also just randomly come up with a new backbone as well. And so just the big picture is, we know inside the cell, we go from DNA to RNA to amino acid sequence to protein. And one of the big goals or hopes in the world is to potentially do that inside a computer. Well, at least it's some of our goals. I guess it's not everybody's goal in the world, but to go from amino acid sequence to a protein structure. But the other in reverse direction is to say, well, can we go from protein structure to sequence? And the idea is, can we use potentially computers to be able to do that? And we know proteins are just a long string of beads. And we have beads that are oil-like and water-like. And the idea is, if you throw this beads of string into a bucket of water, what will happen is, as you probably will guess, is that all the oil will come together and all the water will come out on the surface. And this turns out to be one of the main driving forces of folding. The reason why I'm showing you guys this, even though it may seem somewhat childish, it's one of the things that we'll be later on evaluating to understand, does our model understand folding or not? The idea is, if the model understands folding, then when you design a structure, it should be potentially putting hydrophobics on the inside and hydrophilics on the outside. And it's something that we know with Rosetta sometimes is a difficulty, and you sometimes have to hack Rosetta to actually be able to do this. And so one of the things we'll be evaluating is, does our algorithm understand this very, very basic concept of, you need to put hydrophobics inside, hydrophilics on the outside. And it's one of these driving forces of folding. And in fact, if you look at natural proteins, we see soluble proteins, hydrophobics inside, hydrophilics outside. And transmembrane proteins, it's almost flipped, where you have actually hydrophobics pointing towards the lipids. And it is actually quite an important force. And here's a quick picture showing the landscape of folding. And so the idea is that when you start off as an extended chain, and the assumption is that you can follow this landscape down until you get to your global minimum. And the reason why I thought I'd show this is because it's actually also a really important thing to think about when you do protein design, because often people like to think, okay, I'm designing a sequence for a particular structure, but in reality, what you're really designing isn't the entire landscape. You're actually designing a landscape, not just the structure. And the reason why I'm saying this is important is because, let's say if you designed a sequence and you want it to go to the structure, but let's say if there's an alternative minimum somewhere, you need to really consider the fact that maybe it's possible that you may get trapped in some local minimum during the folding trajectory inside the cell. So it's something that has been sort of missing inside Rosetta, and one of the reasons we do a lot of ab initio folding or forward folding is to say, well, let's design for a particular structure, but then let's evaluate to make sure it doesn't get trapped or there isn't some alternative minimum somewhere out there. So this is just some very basic slides, but I thought I'd introduce them just so we start thinking about the big picture and how it all relates to protein design. So just a quick review how we actually do this inside Rosetta. So in Rosetta, we actually incorporate all these information that I talked about. So besides just the hydrophobic hydrophilic effect, we understand that residues can't get too close to each other. We know side chains have a certain conformation. We know some information about the backbone. We have all this information. And so Rosetta itself actually comes up with a combination of terms of both physics and knowledge-based terms.
Sure.
Go ahead. Sorry, I thought I heard okay. Oh yeah, those of you who are joining in, feel free to interrupt me at any time. I think I often like to talk a little too fast. And if there's no questions, I'll just like finish in 10 minutes. Okay, so Rosetta, I guess most of you probably are aware, but Rosetta has all these terms, and the idea is that if we have all these terms, maybe inside the computer, we could almost simulate some kind of procedure where we start from extended chain and go into a particular natural conformation. And the way we actually do this inside Rosetta is using ab initio structure prediction protocol, and just the exact same way that we have discrete conformations for the backbone where you can be sort of an alpha space, a helical space, or in a loop space. Turns out you could also do this in terms of fragments. So the way we actually do this inside Rosetta is we take the sequence for each stretch of about 3 to 9 residues, we predict a fragment, and then these fragments are assembled in a Monte Carlo trajectory to come up with the structure. And here's a quick animation of that happening. Here we're randomly recombining the fragments, and we have a potential that is described using the knowledge-based and energy-based terms. And so then the hope is that by the end of the trajectory, you have reached the global optima. Okay, but it turns out it works to some extent. So that this kind of thing works like for proteins maybe of length 40, 50. Sometimes if they're highly idealized and the loops are very short, you can sometimes maybe get away with 100, maybe 150. But we know in nature that this is actually very, very hard to do, especially if your proteins are multi-domain and they actually form these gigantic complexes. And so even though this seems like we may have solved the problem, it turns out we're really, really far from solving the problem. And even today, we're nowhere near solving this problem. But we could potentially cheat, turns out. So one way we could cheat is using evolutionary information. So what we could, so here what I'm showing here is a sequence of a particular organism, in this case, the human organism. And these are just little cartoons of different amino acids. And so the idea is if you have the sequence that forms into some kind of 3D structure, you might be able to use some physiochemical intuition to say, well, some of these things should be paired up in certain ways. But one thing you could do is if you actually have multiple sequences, you could potentially start looking at co-evolutionary information. And so one thing you might notice from looking at this plot, this little cartoon here, is that this position is completely conserved. And so that might tell you some information that maybe this is a residue that's maybe in the core of the protein. And then these two residues are covariant. And so that might tell you some information that maybe these two residues should be connected to each other. And then finally, this position is completely variable, which might tell you, okay, this position might be on the surface of the protein. And so when you're just looking at a single sequence, it's a little hard to tell that information. But when you have multiple sequences, you might start to make some interesting inferences. And there's been lots of algorithms developed to try to extract this information from the multiple sequence alignments. But the idea is one of the key information that you could potentially extract is this coevolutionary information. And the idea is if you have that information, you could tell you that these two residues are in contact. And it's because of these residues are so important to be in contact that nature actually optimizes. And then by looking at the evolutionary record, that we can extract that information. I won't go into details of how these algorithms actually work. This is something that we've covered in the past. But I'll just quickly show you that. So this is a protein structure represented as a contact map. And here, these residues here, this blue part, this red part is interacting. And so I could potentially highlight them as dots and this contact map. And I thought I'd quickly show you this contact map as I'll be showing these quite a bit. But the idea is that we can go from N terminus to C terminus, N terminus to C terminus, and then contacts are represented as gray dots. And here are actually overlaid predicted contacts based on the coevolutionary information. And so you could imagine if you had no idea what this structure looked like, and you had only those blue dots, you could potentially throw them into some kind of simulation algorithm and snap this protein in place. So you could start from the extended chain, put a bunch of rubber bands and try to snap it into place. And this was sort of a very exciting breakthrough that's been happening in the last decade or so. And we've been exploiting this, and also Chris Hender, who I believe is on the call, has been exploiting this information, and Deborah Marks as well. But there is one thing to consider though, and that is that whenever we see residues that are in contact or covariant, it doesn't actually mean they may be in contact directly. And what I mean by that is we often assume, okay, if they're covariant, this string kind of folds on itself and it makes these contacts. But you may also have this other scenario where there might be two copies of the protein, and those two copies are in contact with each other. Another scenario is maybe those contacts are not actually, those residues should not be in contact, but maybe they're mediating some kind of ligand or metal, and they may be all covariant with each other, but they should not be that close together. And finally, in any particular given structure, you may actually have multiple conformations. And so maybe one structure has one set of contacts, one structure has another set of contacts. And the reason why I'm bringing this up is because it's actually very important to know this, because if you try to predict the structure here, and if there was just some random noise, turns out it doesn't really hurt any of the algorithms. But if you have systematic noise that actually comes because of these particular scenarios, like you say have homoligomeric contacts, let's say just suddenly pops up out of nowhere, just by randomly filtering things, you don't really get rid of that signal because it's actually real. And so that signal actually hurts structure prediction, makes structure prediction really, really difficult. And just to give you a quick review, there's been quite a lot of people that try to solve this problem. One of the approaches was by Willie Taylor, where he's like, you know what, actually, I could probably just enumerate all possible folds or a database of folds and say, okay, which structure matches that fold? Another approach, DCA fold or EV fold, saying, well, maybe I could just take a subset of the constraints and try fold up the structure by doing effectively like an MD approach or a minimization, and then score with all the constraints. And so the idea is like, if you maybe hide a fraction of those contacts, maybe just by chance you would have removed some of those things that are not gonna get in the way. And then another approach pioneered by David Jones and David Baker's group, and I was partially involved in this, is saying, well, maybe we can use programs like Rosetta or FoldFrag from David Jones and just do a huge amounts of sampling, like literally have hundreds of thousands of computers running for days, sampling lots of structures, and we don't force any of these contacts, just code them as ambiguous constraints. And it actually turns out it works really, really well, but the problem is that it's very, very compute intensive. And so to get around this problem, people have been trying to see, can we somehow potentially filter these contacts? So the idea is, as far as the neural network world is concerned, is you could technically take Rosetta and filter the contacts, or you can use a neural network and filter the contacts. And the idea here is that you could potentially use some kind of a black box model, and this was work primarily done by David Jones, Jimbo Hu, and DeepMind most recently. There's other people involved, so I apologize if they're included in this etc. component. And But one thing that was quite different though, so people have been doing this sort of denoising of contacts for a while, but one thing, the innovation of DeepMind and Jumbo Q was that, well, saying, well, instead of actually predicting just, is it in contact or is it not in contact, what we could do potentially is predict a distribution of distances. So the idea is like, instead of just predicting if these things are yes or no, where you sort of have a single cutoff, you could say, well, what is their distribution? And the nice thing about doing that way is that you get a bit more information, and so then you might be able to get a better structure. One thing that was sort of innovation from Ivan Onishchenko, who developed TR Rosetta, is to say, take it a little step further and say, well, instead of just predicting the distance between the C beta terms, we could predict the dihedral, C alpha, C beta, C beta, C alpha. Then you could also predict the angle and C alpha, C beta, sorry, C alpha, C beta, C beta, C alpha, C beta, C beta, and then also this dihedral here. And it turns out if you actually have all this information between every pair of residues, that's actually all the information you need to perfectly reconstruct the backbone. Even if you bin up the information, you can reconstruct the protein within 0.2 angstrom RMSD. And so the idea here was like, instead of having C beta and then hoping that you will get the rest of the backbone correctly, if you switch to this representation instead, then if you can predict this representation perfectly, then you can perfectly reconstruct the backbone. So that was the innovation of TR Rosetta, and the word TR comes from this transformation of this 6D information. Any questions up to this point? Let me check if there's any. Okay, all right, I'll just keep going. Yeah, if anybody has questions, feel free to interrupt and ask any questions. So the reason why I'm bringing up all this information is because this is the original motivation for TR Rosetta and why we built this algorithm. But now the idea is, can we use it to start predicting and maybe potentially do design? And so this is a quick outline. I won't go into the details of the actual neural network. This is something that I covered before, but if you guys are curious, this paper goes into great detail. So the idea is we start with a bunch of sequences and we pass the first sequence to the network. We pass the conservation information to the neural network, and we also pass the coevolution information to the neural network. And the way we pass the coevolution information is by taking the inverse of the covariance matrix, which is an additional shrinkage term. And so the model actually sees all this information, predicts the 6 degrees of transformation, and then that gets passed to Rosetta to create the 3D structure. But it turns out, which was actually quite surprising for us at the time, was that if you actually remove the coevolution information and replace the sequence and the conservation with just the first sequence, so just literally set all this to 0, that TR Rosetta still appeared to be able to predict some of the structures. So it wasn't able to predict all of them, but it was able to predict some of them. But the part that was probably the most surprising was that, I mean, maybe not too surprising, but at least one thing that was consistent was that it was actually very, very good at predicting de novo design proteins. So all the de novo design proteins from David Baker's group and the Rosetta community, we could actually really, really easily predict all those structures. And here, oh, go ahead.
Could you say, like, I don't know if you know the number, but like, roughly speaking, how much of a hit do you take? Like, if you had to do this across all of the proteins in the validation set, like, what's at the M score of delta?
Actually, here we go. Let me just skip a little bit further. Oh shoot, sorry, I don't have the delta values, but
this is just as
yeah. I'll have to dig that up. I don't remember off the top of my head, but you do hit. There's a pretty big delta for natural proteins, but not for single sequence. Sorry, from design. Sorry. Actually, sorry. Let me step back a little bit. I think I kind of lost my train of thought there. So surprisingly, it worked for single sequences, but it appeared to only work for design proteins. And also touch upon this topic, why that's true. But maybe not too surprising that it worked on single sequences, because it turns out the way we actually train this model is a little bit different than what other people usually train these models with. So what happens is during the training procedure, we take the multiple sequence alignment and we actually randomly subsample sequences from that. And sometimes we might sample one sequence, sometimes we might sample 1,000 sequences, sometimes we might sample 2 sequences. So every single time we sort of effectively do this data augmentation procedure where we try to subsample an alignment and predict it. And so the model itself has actually been trained effectively to deal with everything from one sequence to thousands of sequences. So for that reason, it may be not too surprising that when we finally gave it one sequence, it was able to do something. But the thing that was kind of surprising, though, was that it worked for de novo designs, but not native proteins. So this right here is showing all the de novo designs. And actually, this plot might be a little easier to understand. So this is TM score. The larger the number, the better. This is how close you are to the correct solution. And when we look at the de novo design proteins, they are here. When we look at the native proteins from nature, they are here. The thing that was actually kind of also interesting was that it's not only that they were just incorrectly predicted, the probabilities from the network were actually lower. So if we go back a little bit, one thing I maybe forgot to emphasize is that even though we're predicting sort of these distances, we're actually predicting a distribution that is, you can think of it as a probability. And so whenever we would give it a natural protein, it's not like it would incorrectly tell you that it's very, very confident and then it turns out that it's wrong. If it's not confident, it is also most likely going to be incorrect. So the neural network was actually telling us, yo, we are not confident here. And it turns out that that's also matches the precision of the predicted structures. And then when the network, and it turns out for de novo proteins, it was actually very confident and also very accurately predicted the structures. And so this was sort of like what led to the idea saying, well, if the model can do predict de novo proteins, maybe we can make more de novo proteins. And just to give you a quick example.
Oh, you go on. So this is not quite the question I was asking. So I guess my question was, if you just look at native, and you look at with the MSAs and just single sequences, do you know what the distributions look like? Like, is it like
sorry. So I don't have the plot on me, but yeah, all these guys get actually shifted all the way up to here or so. Yeah.
So it would be kind of like almost. Yeah.
Yeah. So I guess what I should have. Oh, sorry. They would have been shifted up to here. Yeah. I could send you a plot offline. I believe we have something like that in the paper, but I don't remember at the moment.
Okay.
Good question though. It's something that, but it does actually lead to some interesting sort of scientific questions, like why would de novo designs work better than, and one hypothesis that maybe what's happening here is that de novo proteins are very, very stable. Where every single residue is just perfect in every possible way. While for natural sequences, they're all very, just one mutation away from completely not working anymore. And that's probably part of to do with like the protein needs to be able to be adaptive, be able to accumulate mutation to sort of be able to survive in new environments. Maybe to some extent needs to degrade at some point. It needs to function. It needs to bind to ligands and so on. And so the sequence is not very just optimized for the structure, it's optimized for function. And so for that reason, it kind of makes sense that maybe if the sequence, all it does is build that structure and doesn't have any utility, then maybe TR-Rosetta can predict it really well. But if this sequence has a utility, like half the residues may be suboptimal for structure, but maybe optimal for function. But nonetheless, for de novo designs, it was kind of cool that it appeared to be able to predict it. But the part that got us really excited is that we could actually take the proteins, and this is work by Ivan mostly in this case. So what he did was he took the proteins and he tried to mutate every single residue to all 20 other amino acids. And it turns out even with a single, like you literally change one amino acids, the predicted contact map actually looked a bit different. So just to give you a quick example, so if you were to mutate this P into K, you can see that this whole helix interaction has now shifted.
So one question.
Go ahead.
So, I don't know about others, but I actually experienced like 20 seconds of lag while just 30 seconds ago. So Ivan mutated each individual amino acid and then was able to understand the consequent change in structure.
Correct. Yes. Yeah. Exactly. So the question was, So one of the questions was, okay, does the neural network just look at the whole sequence and then decide what the structure is, or does it actually consider every single residue when it's predicting this protein structure? And one thing we found was that if you actually mutate, so in this case, if you mutate this phi into k, sorry, I should probably explain this. The red dots are actually contacts extracted from the PDB structure. In this case, it's one of the de novo design 6DG6. And if you start to mutate any of the residues, you'll start to see that the contacts of the structure no longer match the contacts of the predicted structure. In fact, you can even take this to the next level and mutate every single residue at every single position and construct effectively a PSSM. And one thing that probably is not too surprising was that in the core of the protein, these are the residues that were most changing the structure, and so we would predict there are the least tolerant location.
Is this?
And so, oh, go ahead.
Sorry, I'm just a little bit curious. Are there structures that exist, like the actual structures for each individual one of these mutants? It seems like a lot of structures.
So these are all predicted structures for each mutant, and what we're doing is we're quantifying the differences in each of those predicted structures. Okay. In this case, we're doing it slightly simpler by not necessarily going all the way to structure, but just considering the contacts, saying how different are the contacts from the wild type.
They didn't look too dramatically different if we go one slide back. Sorry for interrupting on this.
No worries. Actually, so if you unfortunately I can't zoom in here, but if you stare at these right here and stare at these here, you can see even though, yes, the helices are predicted to be packing against each other the same way, the contacts are completely off there. So there's at least one whole maybe turn different from one to the next.
So maybe asking a question on that one.
Oh, go ahead.
So phenylalanine 39 then is a solvent exposed hydrophobic, which is this?
Oh, sorry. So I think I probably should have emphasized why these two particular plots in general. So the idea was we wanted to Yvonne was trying to show that if you mutate something on the surface, it doesn't appear to affect the structure, but if you mutate something in the core of the protein, it now appears to be affecting the structure based on the predicted information.
I guess by my eye, I don't really see a difference between the two contact maps that are there.
Good question. Let's see if we can zoom in a little bit. Is that a little easier to see?
I see.
So if we focus on this particular helix where that mutation occurred, the red guys are the contacts. Oh, sorry, I just realized that this P should be not a P, but it's too late for that now. So if you notice, these contacts don't overlap, and if we scroll back to here, you can see these contacts overlay with the gray. So in this case, it's a very, very subtle change, but it now tells you that these residues, this helix and this helical packing will be slightly disrupted or are changed. So it's you'll just maybe rotate that helix just a little bit.
Cool. Makes sense. Thanks, Sergey.
Yeah, sorry, I should have.
Sergey?
Yeah. Oh, go ahead.
So this is not really completely de novo, it seems to me. It's a 4-helix bundle. So
sorry.
it should be.
Okay.
It should be. Let me. I have a 2-part question. Very short 2 parts. So it should be robust intuitively to a lot of abuse, unless you start messing with proline and glycine. Can you talk a little bit about that? Like if you take any one position in this bundle and start messing with it, other than proline and glycine, you should be good for a lot of mutations, I think. And maybe aspartate.
So this right here is the total scan of each position at a time. So if we look at proline here, yeah. So this is a helix right here, and so if you put prolines here, or here, yeah. Sorry, I realized that this is mislabeled. This should not be P. But so if we go through mutate any of these positions to prolines, you can see that it will indeed disrupt it. But if you so these will be positions that are less tolerant. Yeah. And it's a good point. I should probably make sure that this is indeed a de novo design. It was just the design that we picked out because it was one of the David's designs. Okay, so this is sort of what led to the idea of saying, well, if it's possible, so if you think about it, like if it's possible to mutate residues and it tells you that now it's a different structure, you may imagine, let's say if it was the reverse, where let's say you started off with something like this, you can imagine mutating back and sort of fixing the structure. And this sort of led to this idea that maybe what we could do is we can take a 3D structure, convert it into coordinates that we want, and then try to figure out the sequence to get in order to match these coordinates. And so this was the idea of how to do protein design with this kind of model. So we saw that it had some signal, and in Ivan's recent hallucination paper, which I might touch upon at the end, is the idea there was like, well, since we see the signal right away, we don't necessarily have to do anything fancy. What we could do is we could just literally start just randomly change the sequence and do Monte Carlo and optimize the sequence until we get a desired contact map. But for this talk, I'll say that we could actually do something slightly better than that. And this comes from this idea that, let's say if you have a model that makes some kind of prediction. And so in this case, I'm coming up with a really toy example. So let's say you have a model and you want to figure out that what is in this picture, you will tell you, sorry, I'm losing my train of thought here. Let me rephrase that. So let's say we build a model that takes a picture and says, is it a cat, dog, or is it a fish? And so what we could do is we could train this model by updating the parameters of this model until whenever it sees a picture of a cat, it actually tells us that it's a cat. Every time it sees a picture of a dog, it tells us it's a dog. And the way we actually optimize this model is we literally say, okay, you compute the partial derivatives via the gradient function that says, how do I change these parameters until this loss is minimized? And then this is just the basics of how you train a model. But it turns out that with a single line of TensorFlow or PyTorch, you can say, actually, let me just give you a completely random thing. So let's say we learn this model to do this classification, and now we give this model something completely random, like either all zeros or noise. And then you'd probably expect if it sees all zeros or noise that it'll say, well, it's 33% chance of cat, 33% dog, 33% fish. But let's say we really, really wanted this picture to be a cat. So what we could do is we can tell the model, we can backprop to the model and ask the question, how do we change the input to make this model tell us that it's a cat? And so the idea is if you take the gradient, it will be sort of this inverted cat. And just to give you a quick example, so this is an idea that's been explored since at least 2015, maybe even before that, where people try to sort of take models and try to understand what are they learning. And so the idea is you could start with some random noise and you say, hey, I want a banana. And then you keep changing this noise until the category of banana is maximized. And the nice thing about doing this is then you can start seeing like, hey, what is the model actually learning? And here's another kind of cool example where right now the classifier says this is a banana. And what I could do is I could learn, or I could train the model to return a psychedelic sticker that I could put anywhere. And now this classifier thinks that this thing is no longer banana, but now is a toaster. And this is something that might become more relevant as we enter the apocalyptic reign, I suppose, where you have drones flying over your head and you could put a hat on and maybe you'll be able to avoid some drones. But the reason I'm bringing all this up is because we could technically do the exact same thing with protein design. And just, sorry, one more example. So in this case, what I could do is I can give the neural network a bunch of clouds, and then I say, what do you see here? And if the network says, well, I think I see a pig in there or a fish somewhere in there, well, you say, well, okay, whatever you see, just maximize the probability of everything that you see. And if you just keep maximizing the probability, you'll start seeing actual things pop up. You'll start seeing actually fish. You start seeing little houses. And so using this very, very simple procedure, like literally one line of code in Keras, you can start to update your input instead of updating your parameters. And then you can sort of convert noise into any particular classification. You could design something new, a new sticker that fools the classifier, you can make artwork. And at this point, I'll say, anybody have any questions or concerns?
It does seem like that if you were to be using that example of the banana with the psychedelic sticker to convert it to a toaster, that when you are thinking about a protein sequence and then perhaps modifying it, and so starting from an initial random set of amino acids that you want to then convert into a desired structure, that I mean, if you were given what you've said here, it seems like just slapping on the like figurative psychedelic sticker, like it should result in a lot of problems. It doesn't seem very reasonable that you can just like take this random sequence and then convert it to a desired structure. Given what you've said here, is there a twist that I'm missing?
Right. So no, that's a very good point. So this is something that I will touch towards the end of the talk. I mean, the thing is, yeah, it's easy to flip these models around, and interestingly, people have been flipping these models around not because for design purposes, but actually to try to find holes in the model. And now the question would be like, let's say if we actually optimize TR Rosetta, are we actually designing a good sequence, or did we actually find holes in the model? And this is something I will touch upon towards the end. But it's a very good point. Like we want to know, did we find a hole? Did we like, for example, one example that this Inception blog post, if you guys are curious, can go here, highlights is that let's see, that sometimes you would get things that don't actually quite look like they should. Unfortunately, I forgot to include that example. There was one example where when we were asked to predict a dumbbell, it would always include the arm. And so when the arm is missing, it would never classify it as the dumbbell. And so you can sort of start seeing like things like that. Okay, but Dylan, I will get to that at the end. That's something that we did evaluate. Okay, so and one quick note about this is this whole inceptionism idea was partially developed by a guy named Mike Tyka, who used to be in David Baker's lab back in 2012, 13 or so. And he's been sort of pushing us saying, hey, can we do at some point dream up of new things? And so finally we caught up to his ideas, and finally we're able to start thinking about these kind of things. Okay, so now let's see if we could actually do the same thing with proteins. So in this case, let's say we have a protein structure and we can convert it into a target set of probabilities. There's a slight difference here. So instead of, let's say, going towards a single particular label that we want, we say, hey, we actually want not just a label that says this is a protein, not a protein, but actually we have an entire set of contacts and we want to match all these contacts. So now let's say we start with a completely random sequence. We pass that random sequence to our model, and the model gives us back a predicted set of distribution as well. So these are the contacts that it predicts. These are the contacts that we want. And we see that they're quite different. And so what we could do now is we can compute the difference between these two contact maps and construct some loss function that says how different are these categories from these categories. Then we could take this loss and literally just say, give me gradient, or how do I, which tells you how do I change my input to minimize my loss function. And so now if I have this gradient, what I could do with this gradient is apply it to this random sequence. But instead of applying it directly to this random sequence, it turns out you have to do a little bit of a few trickery, because what happens is you want the network to see the categories. You don't want it to see some soft sequence, because it could actually find a hole where it, like say 0.3 this, 0.1 that, 0.5 this. And so what we actually do is we have sort of this hidden variable, which I'm calling PSSM, and we're updating this hidden variable. And then from this hidden variable, we sample the sequence, or we take the argmax of that sequence. And then taking the sequence, we go back to the neural network, get new gradients, and then we send these gradients, update this extra variable, and then once again, and we just keep doing this until this difference is minimized. And this idea was first described back a while back, and here's a paper if you guys want to check it out. But also a few related work. There's another group at University of Washington that did actually something very, very similar to what I just described. So if you guys want to check it out, here's a link to that. Also, folks and Jennifer Litzgarden talked more generally about this idea, saying, hey, how can you take models that predict things and then flip them around to do design? But also along those lines, other folks have taken structures and conditional structures, try to predict sequences. And these are two papers that I'm aware of. But if anybody has any other references, anything to do with protein design, but condition on protein structure, please send me an email. If we excluded you, it was not on purpose. It was mostly because of our ignorance. So please send us your links. We'll include them in the next draft. One thing I wanted to point out about this paper is they also had a pretty cool idea where what they do is instead of just updating the gradients, they would actually try to normalize per position. And the idea there is that by normalizing per position, that maybe you'll prevent the model from getting stuck in some local minimum. Because here, what can happen with the approach that I just described is that as you keep updating this, you may quickly get stuck in some local minimum and can't go really further. So the idea is like, instead of before taking the before sampling, you would first normalize per position. And so it's something that we haven't actually tried yet ourselves, but definitely worth checking out as well. And now at this stage, what I'll do is I will switch and show you guys a quick demo of this procedure. But before I go there, does anybody have any questions up to this point? Or concerns or comments? Actually, if somebody just says one word, that'd be great, because just so I know that I'm actually still connected online and you guys can hear me.
Word.
Word.
Awesome, thank you. All right, so I don't know if you guys have already tried this, but if you go to this link here, all this procedure that I just outlined is actually directly accessible online. And you don't need to install anything. You can just directly run all the code in Google Colab. So what I'll do is I'll go ahead and connect. And all the information, like the model themselves, the utilities, an example PDB file, all the models themselves can be downloaded here. And I'll go ahead and run those code really quick. And so the general idea is we're gonna go ahead and run the code to do this really quick right in front of you. And so the first thing we'll do is we'll take a PDB structure and extract these features that I described earlier about. And what I'll do is I'll go ahead and plot those features. And so here you could substitute this with your PDB file by just clicking on this folder and clicking this little folder. Oh, sorry, I think you just need to click on, you can like drag and drop, I believe, into here, and you can open up your own PDB file. And so these are the features that were extracted from the PDB file. Here's a quick demo image showing what those features are. And so what we'll do is we'll actually set up a model to do protein design. One thing I have to point out though is that we actually trained 5 separate models, 5 TR Rosetta models. And when we optimize, we actually backpropagate through all those 5 models. And at the very end, we just take the mean and then we try to optimize all of, backpropagate through all 5. But just for the demo purpose, I'm just gonna use one. Because if I use all 5, it takes about 5 minutes just to set this up. But in the future, if you're actually using the script, make sure you set this to 5. So this sets up the model. And now what I'm going to do is I'm going to tell.
You might explain how do you average across the 5? How do you leverage the 5?
So let me show you. I don't know if you guys want to see code today, but since you asked, so we're literally, let's see if I can find it. This is all the inputs.
If it's going to take you down a rabbit hole, don't worry about it.
Oh, here we go. So literally just looping through, getting the information and taking the mean of that. Probably should have just said that with words.
Oh, the mean in like histogram space or entogram space?
Yes, exactly. Yep. Okay, so this sets up the model. In this case, I'm telling it just load up one model. This is just for demo purposes. And now what we're gonna do is gonna feed in these features that I extracted from the PDB and run it. One thing I should also point out that this return trajectory is just mostly for demo purposes. I would also turn that off. What this does is at every single step of the optimizer, it actually saves all the predictions, and this you could quickly run out of memory if you actually try to save every single step. So this goes through, start with a completely random sequence, and the score is pretty high, and we want the score as low as possible. And actually, for those of you who are curious, this is actually what the score is. So we're taking the features, which is the probability distribution, taking the log of that, multiplying it by the features that we want. And then since there's like 4 sets of features, I'm just multiplying that by 0.25 and taking the mean of that. So this is what the actual calculation is. And so this is now the predicted features. So this was our target features. And as you can see here, we've now came up with a sequence that matches those target features. Here we'll just grab the design sequence. And now the cool part is since we saved the trajectory here, we can actually animate the trajectory and watch it actually optimize live. So the very beginning, you may notice the sequence was completely random. And as you go further through the trajectory, the sequence eventually converges to a particular solution. And here we could also monitor the gradients that are happening. The gradients that are being computed and are being applied to the PSSM at each step. And then now you might wonder, like, okay, that's cool, you got these features. What does a 3D structure actually look like? Normally you would take those predicted 60 features and give it to Rosetta and predict the structure. But it turns out that if these features are perfect, you don't actually need Rosetta. You can actually reconstruct the structure pretty easily. And so I have a quick secret recipe here, which I won't describe, but it can take the 60 features and convert it into a 3D structure. And the secret recipe only works when you have really good features. If there's some ambiguity in the predicted features, you definitely need Rosetta to actually be able to solve that problem. Shuri in my group has been working on trying to take this to the next level. This right here is actually a relatively simple approximation of what she's trying to do. So stay tuned, in the future, we might have something much better. Okay, so now we have a structure that was from these derived features from the design. Okay, so now moving on to the next step and this is just the same animation that I showed just now, but not put on Google Slides, so there's nothing new here. One other example here is, here's a tin barrel, and you can see quite quickly you can design another structure. One other thing that comes along, and this is something that comes out because we're using coefficient information, is that you could actually design not just a single sequence, but you could actually design billions of sequence all in like maybe 30 seconds or so. And the reason why this is possible is because what you could do is, if we go back a little bit, instead of starting with a random sequence and passing that information through, getting prediction, and then trying to minimize the difference between the prediction and the target, what you could do is you could actually start with a bunch of sequences. And this could be as large as how much memory you have in your computer. And what you could do is you could take these sequences, extract one of the sequences randomly from that, take the conservation information, which is the mean of all these sequences, take the coevolution information, pass it to the model, get a prediction, take the prediction, take the difference, backpropagate, get a bunch of gradients for every single sequence, update PSSMs for every single sequence, and then just keep looping this around. So the procedures are exactly the same, but what you get back at the end is not just a single sequence, but as many sequences as you have memory. And the nice thing about doing this is that you now effectively have a library instead of just a single sequence. One interesting thing that we found out is that we needed to add an extra dropout at this stage here, because what happens is, because the model could technically design for very simple structures using just a single sequence, it often just, it ignores all the rest of the information. It doesn't care about optimizing the coevolution information. But if we actually push the model, say, every single feature is important by adding a dropout during training, during the optimization of these sequences, then you will push the model to actually try to come up with the best possible sequence, best possible conservation, best possible coevolution. And just to give you a quick demo of that, so this is now, we're designing a bunch of sequences. In this case, I believe it was 10,000 sequences. And what we're doing is we're monitoring the coevolution signal that's out here. And you can see that by the end of the trajectory, it starts to match contacts that are actually true. So this is the ground truth contacts. So it's actually now coming up with sequences that actually follow the contacts, not just contacts of the target, but also the convolution matrix matches that information. So this was the nice thing, is that we can actually not just back propagate through the model, we can actually back propagate through the mean, we can back propagate through the inverse function, through the covariance function, and go all the way to the sequence, all the way to PSSMs, and update all this information at once. I think what I'll do is I do have a demo here that does that exact thing. And I won't walk through the cells, but you guys can do that in your own time. So this would be coming with a target sequence, setting up a dropout layer, optimizing. You can see all the sequences, and then you can at the end play out the animation. Okay, so that was kind of cool. But now to go back to Dylan's original question is like, how do you know you didn't just hallucinate a toaster instead of a protein? Like, how do you know these sequences make any sense whatsoever? And that's actually a really important question. And that's literally part of the reason we wrote this paper, because I mean, the idea of just backpropagating is very simple. Anybody can implement it in like maybe a minute or two. But the question was, do the designs actually make any sense? So like, do they make any sense? And so to answer this question, what we did was we took out a bunch of proteins from the PDB that were not in the training set. And for every single one of those sequences, what we did was we designed a new sequence for it. And then for each of those sequences, we computed the statistics for it. And just to give you a quick example, we took all the natural proteins from the data set. And what we did was we looked at every pair of residues that are interacting with each other, in this case, within 4.5 angstrom RMSD of any heavy atom. And we asked the question, for pair of residues that are interacting with each other, what are the amino acids that you actually see? What these heat maps are representing is the probability of amino acid A or B divided by probability of A times B. So blue means that it's observed higher than you would expect just by chance, and red means it's lower than you'd expect by chance. These are scaled from -1 to 1. And so what we see with native proteins, which makes sense that it puts hydrophobics interact with hydrophobics, and you see maybe a little bit of ionic interactions here. And when we actually did the same thing for the design sequences, we actually see nearly the same signal. So it's not perfect, but it does seem to understand that you need to put hydrophobics next to hydrophobics, and maybe it's exaggerating some of the signals a little bit. But here you can see, oh, go ahead.
Forgive me for interrupting once again, but it seems like the idea here is, you're assessing just really basic, like, is this a reasonable protein sequence given the basic biochemistry? So were they characterized? Like, were they subsequently were there experimental assays done on each of these individual design sequences?
Not for these particular backbones. Another paper, which I might touch upon if we have time, it did actually go all the way through and expressing these proteins and doing CD measurements to confirm that these things are stable, but not these particular designs yet. Right now, the only thing we were kind of curious about was just like, okay, did it find a hole in the model? Like, did it just come up with some random sequence that makes no sense? Or is it actually giving you back something that makes sense? And so as far as the pairwise potential is concerned, it appears to be returning a pairwise potential that is very similar to the native pair potential.
I'm sure you've heard of this, Sergey, but I mean, one thing you could potentially do, especially if you have a protein with a large MSA, is you could compare the sequence that's learned versus the sequence versus.
Oh, you mean like a sequence recovery or something like that? Yeah. So you could definitely compare it to the sequence and see how well the sequence matches. One thing I should mention is that every single time you run this algorithm, especially for MSA design, you actually get a very, very different MSA, but they are on average about 10 to 20% identical to each other. So it is definitely, so you can almost think of this as doing like a protein family design where you design an entire new protein family. But as we know, for any given structure, you can have completely different set of sequences that have very, very little similarity to it. But if you run this algorithm many times, thousands of times independently, sometimes you actually find a population of sequences that are getting closer and closer towards the wild type distribution of sequences. But the nice thing about here is that we're not just simply recapitulating the sequence exactly. We're actually finding a completely de novo sequence, I would claim. But then of course you could ask like, oh, is this de novo sequence just a hole in the model? And so what I'm trying to demonstrate here is that no, the sequences, even though they may appear quite different from the native sequence, they actually have, they are capturing, they do understand, how do you say, the pairwise potential. And to show you two more plots, they also understand like which amino acids have preferences for which secondary structure type. So just to give you a quick example, if you have a proline, you'll probably have a coil, and we see the same thing with a sequence. And here's the statistics matching the two. So it does understand like certain amino acids have certain preferences for certain propensities for certain secondary structures. But the part that was actually quite very cool for us was that it also understood depth, this hydrophobic hydrophilic effect that I talked about at the very beginning of the talk. So if you look at each amino acid, so these are all the different amino acids, and you ask the question, on average, what is their depth? And for those of you who are not aware, depth is a calculation that tells you how far away you are from the surface. So like, for example, this guy has a very large depth, while this guy has a very small depth. So this number, you could almost think of it like how far away you are from the surface. Technically, the way it's actually calculated is like, what's the closest water to you? And what we find, which was kind of cool, is that TR Rosetta does indeed put hydrophobics on the inside and hydrophilics on the outside, with the exception of prolines and glycines, but that's also found in nature as well. But the part that was what made us really excited, though, is that it seemed to have fixed one of the long-standing problems in Rosetta. So one thing is, if you do design with Rosetta, not using like any layer design, not using any hacks, just straight up default Rosetta, what we find is that Rosetta puts way too many hydrophobics on the outside. And one of the hypotheses why this is true is because Rosetta only sees that one structure, and it's only when you consider the entire landscape of folding that you would realize that that's a bad sequence. And so the idea here was that because TR Rosetta appeared to be putting less hydrophobics on the outside, maybe that's an indicator that it has some understanding of the landscape. And just to tell you what I mean by that, and so to answer like the big question is like, how is this different from Rosetta? How is this different from, let's say, training a neural network that goes from structure to sequence? Like, what are we doing that's special, or maybe special to some extent? And I guess the answer to that question is that maybe we are seeing, what we're doing is that we're seeing these alternative conformations. And here, this is a cartoon that was drawn by Christopher Norn. And so the idea is Rosetta, the current Rosetta, we would say is effectively this purple eye. And what it's doing is it says, okay, I have a target structure. So this, let's say, is the target structure. And it's gonna, actually, sorry, I had that flip. Let me restart, apologies. So Rosetta is like this green eye. And so Rosetta, what it's trying to do is it's trying to find a sequence to match a particular target structure. So the x-axis is a particular target structure. But as you're digging deep, 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 deep into this minimum, what you may find is that there's actually an alternative structure that sequence can adapt into. And the problem with Rosetta is that it doesn't see this alternative structure. It only sees that structure that it's optimizing. And it's only when you do forward folding, when you do these giant folding simulations, that you'll realize, hey, actually the structure can adapt into a completely different conformation. And so what we're claiming is that by taking this prediction model and flipping it, inverting it, that our design protocol is actually seeing the landscape. And so it's, when it's digging deep, it's actually considering all the other points along the conformation. Here's a quick 3D image made by Basile, one of the co-authors, to illustrate the point. So the idea here, this is Rosetta design here, and this is TR Rosetta design. And so the idea is when you're when you're minimizing the energy given structure, and let's say this is the target structure you're going for, your eye, which is Rosetta, only sees this, and it doesn't understand that there might be an alternative conformation out there that the sequence might prefer. While here, what we're doing is we're maximizing the structure, the probability of structure given the sequence, and we're actually seeing the entire landscape. And so this is why we kind of are quite excited, because we're not just making Rosetta faster. We aren't just neuralizing Rosetta. We are potentially explaining or adding this whole extra information that Rosetta does not know about until you do forward folding. So in some ways you could think about it as we're effectively doing forward folding at the same time as we're doing design. So let's say if you wanted the same thing as here with Rosetta, you would have to take a step along the Rosetta gradient, and then you would have to do forward folding, take another step doing forward folding. And this right here is at the same time optimizing the sequence and doing forward folding at every single step along the line.
Do you think it's happening just when you do MSA optimization or even just single sequence optimization?
We think for both. But as Dylan pointed out nicely is that we actually haven't done any experiments with neither the MSA nor the single sequence design. We've done that for the hallucination project, but not for designing for particular backbones. So that's something I guess will be kind of cool to explore in the future. So right now, this is all just pure hypothesis. And so, but the reason why we're so excited is because we're thinking that maybe this is capturing something Rosetta does not, and not just trying to make Rosetta faster or neuralize Rosetta. Okay, so any questions or concerns up to this point?
This is super exciting. So maybe this is the section you're about to get into, but just thinking, it's shown that you can create sequences for known backbones, but in de novo protein design, the first step is creating a backbone with no sequence.
Yep.
So if you use any of the sort of traditional de novo design backbone generation methods, and you then take those backbones, plug them into TR Rosetta, and get sequences for proteins that have no structural homolog.
Yeah. So I mean, one thing, unfortunately, Chris, is that you could always. It seems like you can almost always find a sequence for almost any backbone, and that's a little concerning. Well, it turns out that's not completely true. If you give it complete crap structure, it can actually find a sequence to match that structure. But for de novo designs, it could always give you back a sequence, which is kind of cool. But also there's always the concern that maybe we're overfitting. This is part of the reason why I was really excited about adding this dropout here, is because you could also imagine that maybe what could potentially happen is that because if you're doing this gradient-based approach, if we go back a little bit to the original structure here, if you're doing this gradient-based approach, you can imagine that it might find the bare minimum sequence needed to predict what you want. And then after that, it doesn't really care about the rest of the amino acids. And that is definitely a concern. And so it's something that still we need to test to see if that's a case or not. One thing I could do, I think I have a little bonus slide at the end, which I can jump really quickly, is just to illustrate why I'm so excited about this MSA design approach is if you actually run the single sequence design approach 10,000 times independently and design a whole bunch of sequences, and then you take those sequences and you throw them into, oh, go ahead, sorry. Oh, okay. So if you, was there a question there? Okay. I hear like a robot voice, but I'm not sure if that's a person or just static. Okay, I guess I'll continue then. So the thing is, like, let's say if you take that single sequence design approach and you design hundreds of thousands of sequences, and you take those sequences and you throw them into a coevolutionary algorithm. The thing that we find that's actually kind of interesting is that it doesn't appear to have all the contacts. Like, so this is the backbone that we're designing for. These are the contacts extracted from the backbone that we're designing for. And all the sequences that were designed appear to be only making a subset of those contacts. Like, it's making these contacts, these contacts, these contacts, but does not appear to be co-evolving these contacts, or at least the sequences don't appear to be co-evolving. I mean, there could be a caveat, maybe these positions are completely conserved and that's why they're not covariant. But it did make me a little worried, thinking like, okay, maybe what happened was TR Rosetta just came up with a sequence that makes some of those contacts, and that was enough to predict the structure, and it was happy, and then to not try to optimize the rest. But also on the scientific point, was kind of curious because maybe these are like the key motifs that are important for the folding of that structure. Maybe the rest of it could be greasy and we don't really care about the cohesion there. Now, when we do the MSA design approach, on the other hand, all the sequences that come out do actually have the contacts. And this might be slightly cheating because it's using the coevolution. So of course it's going to put context there because it wants to use that information. So that's part, it still needs to be sort of validated with experiments. But there is some concern, or I'm not sure if it's a positive or negative concern, that maybe you're finding a bare minimum sequence to fold in that structure, and maybe it's not good enough. And maybe by introducing, like one thing I'm thinking is maybe we also need to have a dropout, not just for multi-sequence design, but maybe also a dropout for single sequence design. Then what you do is you push the network to really try to optimize every single position for that fold instead of finding some bare minimum signal. Okay. And I guess we are pretty much out of time, but for those of you who are interested, I could touch upon this hallucination design component of the talk, but there's no pressure to stay if you have things to do. But I guess at this point, what I'll do is I'll stop sharing and say hi to people if anybody wants to say hello. Hi, everybody. Hey, Chris. Any questions or concerns? Anybody have?
Great talk.
Thank you. I guess, okay, so if anybody needs to run, I won't be offended if you leave at this point, but for the rest of you who are curious, I'm happy to keep going. The rest of this probably won't take more than 10 or 20 minutes. I don't have too many slides. I just wanted to show that so this is a question that Chris Ball asked earlier. Could we somehow...
Sergey, can I ask you questions before you go to the hallucination? This is Per speaking.
Of course.
So I think a fantastic result, you know, that very impressive. But I'm actually wondering a little bit about native structures and why you have these issues. Have you tried to actually reconstruct the trajectories and then see if you start to capture some of these primordial motifs that you would actually anticipate during evolution of some of these folds?
No, that's something I haven't analyzed. I think what I have this, so one of the things I forgot to say is one thing that I really like about this MSA design approach that I was proposing is that we know with multiple sequence alignments, we could predict the natural proteins. And so it's possible that maybe with multiple sequence alignments, we could now start to target that region of space that, that a single sequence cannot touch upon. And so the idea would be that maybe when you have a single sequence, you could predict, you can design structures that have ideal folds. But if you want to go into non-ideal zone, then you need the coevolution contacts. And by doing a multiple sequence design, then you're going to start going into this non-ideal zone. So that might be a way to address the problem of why native sequences cannot fold. And we have a way to incorporate that potentially. But of course, all this is just theoretical. So.
And then a last comment to your trajectories of the folding, which was also quite interesting. I was wondering, have you looked into some of these, I think they're termed high energy NMR experiments from Louis Kay, where they actually start to capture these intermediate states that you actually claim at this point that you can actually design against?
I didn't show any figures on that, but one of the co-authors is looking at that actually. Maybe in the next version of the preprint we might have something like that. So we are looking at cases where the same sequence can have multiple folds, or maybe with a few mutations go from one to the other. And one of the questions is how sensitive the model is to be able to capture those kind of things.
So just to comment a little, so in theory, you should be able to, if you did this on an intrinsically disordered protein, right? That should start out with having, you know, very shallow energy wells, and then you at some point will get to a folded state, right? Have you tried to look at any of those proteins to see if they cluster or behave differently during this design process?
We haven't tried that, but that's a really cool idea. We'll have to try.
Okay. So last but not least, and that's regarding the Rosetta energy stuff here, because have David or you or anybody else in the energy function community started to actually look about how to adjust like reference energy or negative state design into the design process of proteins?
To some extent, maybe Chris Ball could comment on that since he's a protein designer. Chris?
Sorry, I was a little distracted there. Could you say it one more time, Per?
So what I'm
Well,
saying is that
while you're thinking, could I just ask who was asking the question, because I try to understand who's talking, who's saying what.
Oh, sorry. Let me rephrase. So Per was asking the question of, did we try to
just ask who was asking the question? Sorry to interrupt. Who was asking the question so I can follow?
Per was asking a question. Per Grayson.
I just raised my hand, Chris.
Okay, thanks.
All right. So my question is that we know that there's this multi-state design problem once we start to do proteins folders. And given the hypothesis by Sergey, what he's shown here in the lecture, right, then we should actually be able to actually design against those states as well as with the physical as well as statistical potential. So my question is that something we try to incorporate into these sampling techniques in Rosetta, or do we just say that the ML version is just better?
So they're in the, I guess I could say what we're currently doing on the ML side. And actually, Chris Paul, how about you go and tell us what we're currently doing before I say what we're trying to do?
I mean, there are multi-stage design protocols, right? Or multi-state design protocols. So you can consider one other state if you know what it is, and you can minimize the energy for one state and maximize for the other, right? This was something Andrew Lieberfey developed a few years ago, and they used to make bispecific antibodies in Brian Coleman's group. But I think the crazy thing with it, at least if this works the way that Sergey is hypothesizing it does, is you don't have to explicitly know what the alternative states are, right? It just sort of minimizes hypothetical alternative energy minima without explicitly enumerating them. I don't see how that would be possible to do in Rosetta. And if that's actually what's philosophically happening here, I think ML is going to be the only way to do that.
Because just to comment a little bit on that, I really think this is interesting if we can start to deconvolute the unfolded state, because that's clearly something from a physics point of view that, you know, I think Chris Sanders can comment on this, but that was really, you know, the beginning of all of protein physics is what is actually the reference state of a folded protein, right?
Mm-hmm. No, I mean, it's a very important point, and that's like why we're so excited about this ML approach. One thing that we're currently doing, in one of the things that I didn't talk about, and this is, I probably shouldn't say we, because I'm just the code writer, everybody else is actually doing the work, is the idea is to maybe what we could do is, because we have now these statistics that come from these ML models, we can potentially construct a PSSM and give that to Rosetta. And so the idea is that now that PSSM will push you in the right location and fold space. And that's one way to incorporate that with Rosetta. But as far as how people done it with Rosetta before, what Chris was saying was that we do have that information, like we have that protocol for multi-state design. But what I've seen people do in the past is when you do these forward folding trajectories, you could either throw out that design if it has an alternative minimum, or you could explicitly say for that alternative minimum, let's design a sequence that just favors that. And that's been, I believe Nobu was doing something along those lines.
So I understand that's been done multiple times, but what this actually shows is that we could start to get the whole idea about the reference state and the reference energies giving a sequence, which would be quite fancy.
Yeah, no, exactly. That's why the paper was written, because otherwise, if it was just, hey, we made a neural network that does the same thing as Rosetta, then we most likely would not have written this paper.
Can you explain a little bit? How do you think you can get the reference state out of this? I'm not entirely sure I follow.
Parrot, was that?
I mean, if you have your sequence and you start to sample how the, let's say, the free energy landscape of your sequence actually behaves on the different minima, then you will also know which reference state you would design against, right? So if you look at the simulation that Sergey showed, I would assume that you somehow can deconvolute that and then transform that into better reference state than we do right now. Right now, it's just a linear chain.
Mm-hmm.
Does that make sense, Mohammed?
I think pairs maybe overcomplicating it a little bit. I think if I was to give you my, how do you say, naive version of that comment, it would be that, I mean, we're now, because we have this function that goes from sequence to structure, when we have that sequence, we can finally design that sequence for that particular structure, we know it can't go into any other structure. Unless our model just overfit on something, so that could also happen. Okay, well, how about this? I'll show one more thing, and this is going to be relatively short, and then we could maybe continue the discussion a little further. Let me share my screen again. Oops. So during the talk, I described this procedure of going from sequence to structure, and then trying to maximize minimize the difference between the prediction and the target. But now imagine instead of doing this, like let's say if instead of doing this, I just say, you know what, actually I just want to maximize the probabilities. I just want to make as many contacts as possible here. Like just imagine cutting all this out and just saying, I want to maximize all the probabilities. Or you could do it even a step further and say, you know what, actually I know how many contacts each residue has on average. Make sure that I match that target distribution. So if I know that proteins on average have, let's say, 2 contacts per length, I say maximize the probabilities of these guys and give me on average 2 contacts per length. And I don't care what this looks like, I just want to have 2 contacts per length. Or you could even take it a little step further and say, well, actually, I know there's a distribution that when you're close by, you have more contacts. When you're further away, you have less contacts. And so that's exactly what Yvonne did. So what Yvonne did was he said, you know what, I know what proteins look like. I know there's some target average number of contacts at different sequence separation. Let me just keep enhancing that output. I guess, sorry, that picture is not very clear, but let me just keep enhancing this output until it matches some target distribution. And that's sort of the basis of hallucination design. Like we're not trying to design a sequence to match a particular target. We're just trying to design a sequence to match a particular distribution of number of contacts at different sequence separations. And actually, I think maybe the best way to learn about this is to go directly to the notebook. So what we could do is we can come up with a matrix that describes the distribution, the background distribution of different distances, different dihedrals, different angles at different sequence separations. And this is exactly what I'm showing here. So for each position, what I could do is I can say at different sequence separations, so like, for example, at sequence separation of 1, we would expect the C beta distances to be 5. At sequence separation of 2, we'd expect it to be about 7.5. At 3, because you can either be a helix or a sheet, there's 2 peaks here. And at 4, it's going to be here. At 5, it's going to be here. Now imagine populating an entire length by length matrix of all these probabilities, and that's your background. So now if you make that as your target distribution, what you could do now is say, we can, in this particular code, what I'm doing here is I'm saying, okay, I'm going to add in the background. And what I'm going to do is I'm going to design a sequence to match that background. And the way we actually define this function is using KL divergence, where we have our background, which is here. This is the output of our model. And we're trying to actually make it as different as a background as possible per pixel.
So, Sergey, do you mind if I just clarify a point that, you know, it's funny, I talk to you every day. Sometimes I miss something. I'm a little curious why the goal here is to maximize the KL divergence between the aforementioned background and the design. Just moments ago, like what's the sort of framework?
Good question. So actually here, Dylan, I think if I show you this plot, everything will make sense. 1 second, I should have included this plot. I apologize. Okay, so plot features.
Absolutely nothing to say sorry about.
Okay, so this is the background distribution for a protein of 50. So the thing is, if you try to match the KL, sorry, let me rephrase that. If you try to actually match the background distribution is like, oh, there is no contacts. But what we want to do is we want to say, well, we don't want to just, we actually want to maximize the difference. And so push it towards, at the very end of the day, you should match this distribution here. It's a little confusing because it's not done on the average distribution per position per sequence separation. It's done per pixel, and henceforth why this equation and not like why this is a negative here. But we could take this offline. It's a little bit convoluted to explain. I think with some frame examples like that might make a little more sense. But,
Yeah.
okay.
Sorry.
No worries. Okay. So this is the target distribution that we have, and now if we go through the design procedure, and this is the function that we're actually optimizing. This is the output that we got. So this is our designed protein. So now we have both a design sequence and design structure. And now if we actually look at the distribution that comes out, we get this distribution. And one thing you might notice is it doesn't actually match exactly this distribution. The reason why is because this distribution says there could be helices or there could be sheets. But in this case, we only have, looks like sheet proteins or shitty proteins, depending on how you want to think about it. And so in this case, we have only sheet proteins, and so you have this distribution, but not the other one. So we're biasing towards that distribution, but not trying to match it exactly. So it's more like an energy as opposed to matching those probabilities exactly. So now if we actually animate this really quick here, so this is the designed hallucinated constraints. This is the sequence that matches those hallucinated constraints. Here's what the trajectory looks like. So as you're optimizing the sequence and optimizing the match to the background distribution. And finally, if you want the structure, here's what the structure that was designed looks like. And that's pretty much all there is to it. One thing I should point out though is in Yvonne's paper, which I didn't go into too much detail here, they did it slightly differently. They didn't use gradient descent because this work was actually more than a year old. Well, maybe almost a year old now. This is when we were still doing MCMC simulations. So it was not using this gradient descent-based approach. So there might be some caveats that maybe there could be some differences in how MCMC design sequence and structure versus gradient approach. And that's something we're still trying to quantify the difference. That's one caveat to keep in mind, that this notebook that I sent is not necessarily replicating this algorithm exactly, because using, Yvonne did it using MCMC generation, while in the notebook we're using gradient descent, which makes things much faster, but may potentially be finding some other different minimums or so on. But nonetheless, so here is just a quick example of structures that were hallucinated by Yvonne's algorithm. And so you can see here, you can actually hallucinate really, really different proteins. And they are actually really, really different from any sequences in UniRef. So usually E-value of about -3 is where people would say these sequences are about the same as something in the UniRef. But the design backbones, interestingly, do actually look like something in the PDB. So in some ways, you could say it's not, maybe not calling it de novo design might be slightly, how do you say, might not be necessarily the best way to phrase it. Maybe one other way to phrase it would be that we've now learned sort of the ideal rules that are encoded in all of PDB, and now recapitulating those rules in a very idealized way. And so these are just examples of backbones that were hallucinated, and this is just showing that they are indeed, the sequences that come out are quite diverse, the structures that come out are quite diverse, and so one thing we could do is now we could take each of these structures, and first one thing we could do is say, do they make sense? Just like the analysis that I showed before, like do they put hydrophobics in the inside? And we see yes, they do. If we take these sequences and we do forward folding, and we ask, do they forward fold? And we see that every single design, we have a funnel towards the correct structure. And the exciting part is it does not appear to be any minimums. And finally, if you take these proteins and express them in E. coli, and then purify them, run CD, they do seem to match the predicted CD distributions in terms of secondary structure and so on. And
this
also
is all very exciting. Sorry for interrupting. I just feel like
I'll go for it. No worries.
though you have said, please interrupt me.
Oh, no, I'm happy for interruptions. Go for it.
Okay, well, one thing is these experimental characteristics, they bode well for the hallucination project. I'm wondering, what is that, like, we're talking about like this very second, is that taking place? Like, do you think that the designs here and the properties of the proteins therein will translate to the designs from the project that was talked about earlier, or do you think that there will be some sort of like gap in something?
I think so. I actually believe, I suspect everything will be should transfer, with the minor caveat that they were designed using this MCMC approach as opposed to gradient descent, and so it's the same. I mean, I guess the loss is a bit different because we're maximizing KL instead of the cost entropy. I think they will, because if we look at the statistics, so I unfortunately don't have the statistics with me, but they do match, for example. So Yvonne also did the same statistics where he looked at the pairwise potential, he looked at the secondary structure propensity, and he looked at the depth correlation. And for all these designs that were hallucinated have the same statistics as the ones that were designed for specific backbones. So for that reason, I would say very likely. But I mean, if all these fail, let's say at the later stages when we actually get the crystal structures, then I would say the other ones will also fail. So yeah, they will either all rise or all fail at the same time.
So are crystals being generated for the proteins that you are showing right now, this very second?
That actually, I don't know. My assumption is, so this part of the project I was not involved in. I'm just showing the data that Tamuka actually collected.
Oh, nice. I should just ask.
And sorry, Tamuka and Sam. My guess is since they went to all the way to the stage, the next stage would be crystallography, but I don't actually know the answer to that question.
If I may ask, so is it that every single like one of those hallucinated designs would be in a way like a collapse mode for this, for like when GANs are used, right? There's this collapse for GAN problem, right? Is that like sorta in a way each one of those like hallucinated structures is like one of the collapses, one of the modes that it collapsed to? I don't know.
That I don't have an answer to that question. I don't know. I haven't really thought too much about mode collapse myself, so.
I mean, I think it's a bit different. I mean, you don't have a GAN here, so it's not the same phenomenon of like an actual mode collapse. And again, it is, you are putting all your probability measure on this one mode, right? So you are concentrating all your probabilities. So you are extracting a mode, but I think it's a different thing.
Yeah. So I guess
it's definitely kind of a mode selection. Yeah.
No, definitely. Like at the very, like actually, if you look at some of these trajectories here, so this is showing at the very beginning when you start with a complete random sequence, what does the contact map look like? And also, if you look at this yellow, what this yellow represents is that it's very not confident. So that it's very, very unconfident about these predictions. As it gets towards the end, it gets very, very confident. But one thing you might notice is that the structure might actually fluctuate through different, I guess you could say, modes, where it begins at this, and then it goes into this weird state, and then it flips and becomes this. So there is a bit of exploration. Eventually, it settles down onto something. So maybe another way to think about it is like there's sort of a space of all structure space out there, and you're sort of navigating it, but not sure if that's also the best way to think about it or not, because there isn't really like a latent space per se. But it does seem to understand that that contacts need to flow in a certain way, or need to be composed in a certain way in the contact map. All right, I'll stop sharing my screen so we could all see each other and talk back to each other if we want to. Hello, everyone.
Yeah, thanks very much, Sergey. I have to go, but it was very interesting. Thank you very much.
Of course.
We'll talk another day. This is Chris. Bye-bye.
Take care.
See you, Chris.
Bye.
See ya.
Chris.
Sergey, can I ask a question more now that I have you?
Of course.
So you made quite a lot of emphasis on soluble proteins. Have you actually tried to reverse the potential and then look at membrane proteins and see if you're actually able to recapitulate them as well?
We haven't tried membrane proteins. Well, I mean, I've personally not tried, but I don't know if anybody else in David's lab is trying or not. I would think it should work because the model was trained with membrane proteins and soluble proteins. Maybe something as simple as biasing the AA composition